Okay, so we know that the second neuron in series, it ends in these distinct neuron endings, and on those endings we have these swellings, which are called varicosities, okay? These are called varicosities. Now, the tissue in question we know is either cardiac muscle, smooth muscle, adipose tissue, or glands, okay? And we're gonna look at two different neurons because we know that there's two options for this junction. It can be on the sympathetic side, it can be on the parasympathetic side. So we're gonna draw this out and see what both of those look like. Okay, so we have our nerve endings and our effector cell, okay? Now, we also have varicosities here. So this will be the sympathetic postganglionic neuron. I'm just gonna put PG here so that you know that it's the postganglionic neuron, so it's that second neuron. And then on this side will be the parasympathetic postganglionic neuron. Okay, let's put PGN. Okay. All right, now let's zoom in a little bit closer and see exactly what's going on between the varicosity and the effector cell. All right, so we got the varicosity and our effector tissue, which we know is some type of smooth muscle, cardiac muscle, adipose tissue, glands. Okay, I keep saying that just so that it really sticks. Okay, and again, here we have the same thing, the varicosity and whatever tissue this is. We'll just say the effector cell. Okay, so far everyone clear, right? So we have two different systems, the two divisions, and we know that this is the second neuron as it synapses onto its respective tissue, okay? Now, on the sympathetic side, what neurotransmitter is going to be stored in this varicosity? On the sympathetic side. What neurotransmitter is stored here? So if this is a sympathetic postganglionic neuron, what's the neurotransmitter? It's not acetylcholine, because this is on the sympathetic division, so it has to be norepinephrine. Okay, so this has to be norepinephrine. On the parasympathetic side, it will be acetylcholine, okay? It will be acetylcholine. Please make sure you're making those distinctions, right? Sympathetic postganglionic neuron releases norepinephrine. Parasympathetic postganglionic neuron releases acetylcholine, okay? So inside our varicosity, we have these vesicles and they will have our respective neurotransmitters stored. Okay. And I'll just do this just to distinguish them, that they are different neurotransmitters. Ready? Now, we know that the very same steps we talked about for synaptic communication also takes place here. So we have an action potential traveling down the length of this neuron. Okay, don't disconnect that bit. That event is still very much relevant here. The propagation of an action potential down the axon of this postganglionic neuron. When it gets to the varicosity, much like when it got to the axon terminal, it's going to trigger the opening of voltage-gated calcium channels. Okay, voltage-gated calcium channels. And these voltage-gated calcium channels will allow calcium to move into the varicosity. Okay, I hope everyone's following me. Calcium moves into the varicosity, and that results in exocytosis. So we have this vesicle docking on the membrane and then emptying its contents into the synaptic cleft. Okay. Likewise, on the parasympathetic, the events are just the same. So calcium rushes into this varicosity, and that triggers exocytosis such that we dump all of our neurotransmitter into the synaptic cleft. Now, on the effector cell side, there are a few options for what happens here. Now, we know we have a receptor, okay? And we know that that receptor is going to bind specifically to our neurotransmitter. So on the sympathetic side, what type of receptor is this? On the sympathetic side, what type of receptor is this? If it's binding norepinephrine, what type of receptor is it? It must be adrenergic, right? It's going to be adrenergic. That's one way we can describe it. What else can we say about this receptor? What else can we say about this receptor if it's adrenergic? It's metabotropic. I'm going to put that in brackets. It's metabotropic. Okay, let's say I told you that this effector cell was in cardiac muscle. What type of receptor would this be? If the cell in question is a cardiac muscle cell, a cardiac myocyte, what type of receptor would that be? What type of receptors do we see on cardiac muscle? Is it alpha 1, beta 2, beta 3, alpha 2? Beta 1, good. So this would be a beta 1. Let's say I told you it was on adipose tissue. What type of receptor would this be? Beta 3, right? Guys, make sure you're making those connections. If this tissue changes, the receptor will also change, right? Different receptors are found on different tissues. So make sure you're making those connections. Beta 1 is on the heart and kidneys. Beta 2 is on the lungs. Beta 3 is on adipose, okay? All right, let's look on the parasympathetic side. So we know this is adrenergic metabotropic. Let's look on the parasympathetic side. What type of receptor would this be if it's binding acetylcholine? So we can describe this in more than one way. If it's binding acetylcholine, it's a cholinergic receptor. But more specifically, what type of cholinergic receptor is it? I think I'm hearing it. Not ionotropic. If it's not ionotropic, then it's metabotropic. But what else can we say about the subtype of cholinergic? Okay, so this is going to be a muscarinic cholinergic receptor, which is also a metabotropic receptor. Okay, we talked about cholinergic receptors. Anything that binds acetylcholine is a cholinergic receptor. But there were two types. There were nicotinic. Where are nicotinic receptors found? Are they found on the target tissue? Are they found on the effector cell? They aren't, right? They're found on the soma of the second neuron. The first neuron releases acetylcholine. All of the somas of this second neuron will have the nicotinic receptors, but the effector cell has a different type of cholinergic receptor. It's a muscarinic receptor. Okay, and understand the difference between nicotinic and muscarinic. Nicotinic receptors are ionotropic. Muscarinic receptors are metabotropic, okay? All right, so any questions? Any questions on that? Okay, so if you're not clear on this, please go back over that. We did this on Friday, the types of receptors, where they bind, what they bind, the subtypes, okay, what tissues they're found on. All right, so then the second neurotransmitter is released, bind to its specific receptor, that brings about some type of cellular response. Now let's look at what happens after we have our event on the postsynaptic, on the effector cell. There may be excess neurotransmitter at the junction, and so we have enzymes here on the parasympathetic side that will degrade the excess neurotransmitter. Anyone remember the name of this enzyme? We talked about it on Friday. So this is acetylcholinesterase. Acetylcholinesterase. And remember I told you that suffix ASC means that something is being cut or cleaved or chopped. And so this enzyme, acetylcholinesterase, breaks down excess neurotransmitter, but only on the parasympathetic side. Acetylcholinesterase cannot break down norepinephrine. In fact, norepinephrine does not get broken down. Okay, it can be reuptaken as it is via its uptake molecule. Acetylcholinesterase needs to be broken down, acetylcholine, sorry, needs to be broken down by acetylcholinesterase, and then it can be reuptaken. Okay? So let's put our uptake molecule here. And this is where we can uptake choline and acetate. And these are the breakdown products of acetylcholine. Okay, and this is again something we talked about before. There were 3 options for clearing the junction. We can have it chopped up and repurposed. We can have it diffused away as it is, or we can have it chopped up and then diffused away. Okay, any questions? Any questions on this? Any questions? Okay. All right, so I hope this was not too complicated to follow. But this is really how you should be studying. You should be tying everything together. Don't study in silos, right? Question in the back, sorry. Sure, so we can have neurotransmitter diffuse away in its original state. So it can diffuse away as is, that's the first option. It can be degraded and then diffuses away, or it can be degraded and then repurposed, reuptaken and then repackaged, okay? All right, any other questions? Any other questions? Okay, so as we talk about the neuroeffector junction, it's really important to remember the first question that I asked was the 2 differences of this junction from, say, the neuromuscular junction or from a traditional neuron-to-neuron synapse. And just to remind ourselves of those 2 differences, one was the presence of varicosities, the other was this greater synaptic distance. Okay, so don't forget those 2 differences. So those set the neuroeffector junction apart from the neuromuscular junction and from an ordinary neuron-to-neuron synapse. Okay.